Namaskar. It is my pleasure to welcome to this India Foundation podcast Professor Giuseppe Tritto from Italy. He's now in Umbria, in Italy.
Yes.
He's the president of the World Academy of Biomedical Sciences and Technologies. He is specialized in microsurgery, and he has many other fields of interest in the advanced medical field, particularly in the field of nanobiotechnology.
Yes.
He will tell us a little more about it. So, Professor Tritto, welcome. Can you tell us a little more about your activities and particularly your connection with India?
Yes, it was a great pleasure for me to meet many years ago Professor Sujoy Gua, who is an eminent scientist in India in biomedical engineering. He created the chapter of ITT in India on biomedical engineering. And it was a period in which I want to organize a great congress on andrology, biomedical engineering, and rehabilitation. Professor Gua has written a book on the bioengineering in human reproduction and andrology. I invite him. So Professor Gua introduced me to the ITT organization in India. I spent some time with him in Kharagpur and the Institute of Technology there near Kolkata. My first experience was there in West Bengal. And then we move on New Delhi. We start to develop technologies, biotechnologies for human contraception. It was a great program in India on female contraception, nothing on male. So we developed a program and a project that was called RISUG. And after RISUG advanced to create a transient contraception method. We can inject this glue into the vas deferens. We stop it for a certain time, the passage of the sperm, and the people for 6 months or 1 year were have this reversible contraception. It was a very simple technique. I have used these techniques in India with him, with the urologist in India, Professor Singh, eminent urology there, and we do a trial. After many years, this was approved by the Ministry of Health in India. We have met the Ministry of Family Welfare in India, many things. At the same time, with this discovery, we found it that with this type of glue that we inject, we degrade the sperm, but we generate a nanoparticle that are the membrane of the head of the spermatozoa that are nanoliposome. And these nanoliposomes were created by the people themselves, by their sperm. And so on that, we decided to use this nanoliposome to charge with the drugs that can have an effect on the cancer of the prostate. So it was the second step of the study that we do with them. We use the sperm of men to create this nanoliposome. We charge this nanoliposome in vitro with product who can have an activity against cancer of the prostate. We can inject into the vas deferens. And the other discovery was that this type of nanoparticle have a transmission, transmembrane transmission, and arrive into the prostate and concentrate into the prostate. There was never discovered this type of mechanism of the transmembrane fluid passage or molecular passage from the vas deferens and the testes and the epididymis into the prostate, because people focalize only on ejaculation process. So we concentrate this type of nanoparticle, and we're starting the protocol to study the prevention of cancer of the prostate in a human being. I am the leading in Europe of this type of trial that we've done, reversible contraception and nanoliposome, charged nanoliposome for cancer of the prostate. This was.
Thank you for this clarification. Obviously, for a general audience, this is rather technical, but I think they got the gist. Now, with regard to your book, you have expounded a thesis which has recently been confirmed by the Pentagon through the release of documents issued by the Veritas group, Project Veritas in the United States.
I know very well.
And basically what they are acknowledging is what you write in your book, which is that the SARS-CoV-2 virus, in other words, the COVID-19 virus, is a chimera. In other words, a lab-made product, which apparently was jointly developed in the United States and China from bat viruses.
Yes.
Now this, and you give a very ingenious explanation, which I won't go into because it probably will take too long, but you are talking about the furin, which shows that this was a human manipulation of a virus.
Yes. This was what Baltimore called, when he see the first strands of genome of the virus. Baltimore was the guy, the virologist in the United States who created classification or RNA viruses. So a great expert, etc. He thought, oh, this is a smoking gun. This insert of furin is a smoking gun. And now I am preparing a new book on the furin only, that is called the smoking gun, and in which I illustrate all the steps that permit to the Chinese to arrive to build a series of chimeric recombinant virus, starting from SARS-1, a SARS-like, they call it that way, all the group of great SARS coronavirus in bats. They starting after the SARS-1, they created 11 on 14 chimeric recombinant SARS-1, SARS-like in laboratory just to 2008. All these type, 11 on 14, were pathogenic for human cells in laboratory. So it was a long history on that, starting from someone. After, when the American people begin to work with the Xi Jinping, the Batwoman in Wuhan, director, of the laboratory of Wuhan, they started in 2005 with some action to find why the viruses, coronavirus in bats in China, etc., some sort of a general action under the aegis of WHO, United Nations. So the American people begin to work together with the Xi Jinping, financing research. It's happened one thing after 2008, after MERS, fundamentally. It's happened some things that China decided to create a big laboratory, P4 laboratory, the first P4 laboratory in Asia, not only China, but in Asia. And they wanted that to be international. So they asked to WHO to endorse the creation of this first laboratory in Wuhan. They do a long passage to have all the accreditation for that, etc. The plan was done by French people, by Professor Lama in Lyon, who created the project, the laboratory. It was similar, that laboratory in Lyon. There are people of Mérieux, who cooperate,
the great pharmaceutical lab,
who cooperate. They were there, but at a certain moment, when the American people entering the cooperation with China, the Chinese put French people outside. Even if the research people in Wuhan, especially Xi Jinping, have learned the technology to create a pseudo virus, HIV pseudo virus from Institut Pasteur, from the French people.
Sorry, so what you mean by pseudovirus is essentially
fundamentally
virus.
Is a core of the HIV virus who has all the properties except the virulence, fundamentally. But in that way, you can implement the technique of gain of function, that is put some things, etc., to show how you can manipulate the contagiosity and the contagiousness and the virulence of the virus. Xi Jinping do a training in Montpellier. After, in the great laboratory of Professor Rotten in Netherlands, Professor Rotten was the great guru in Europe for the recombination of chimeric viruses interspecies. Right. So she learned also these techniques of combination, etc. So with this cooperation with the United States, the real point, the turnout of the problem is when she published in 2013 that she has discovered the sources of SARS-1 in the bats, in coronavirus on bats. Strangely, 6 months after, a Chinese group of military doctors on the virology, expert in virology, published another paper in China that was translated after. They thought, no, this origin is not true, because it's not the father, not the grandfather, not the ancient of this SARS-1. So fundamentally, in China, the military medical environment in infectious disease said, no, we don't agree on this type of origin. Okay? Okay, strangely, is an interesting thing. But the real things start in 2014 when the President Obama created the moratorium against the research on coronaviruses, influenza viruses, and MERS, because there were two leakages into CDC laboratory in the United States. There was a great fear on that. And Obama stopped all the funds, national funds of research on coronaviruses in the United States. So the problem was how this type of research people in the United States can continue their job. Right. They have done two conferences in 2012 and 2013, the group of Ralph Baric and Mark Dennison, who work in long time on coronavirus pathogenic for human in their laboratory. There are laboratory P3, university laboratory.
Yeah, not P4. In other words, lower level.
No. But you know, close with the military environment in a certain way, and they transfer all this type of research in the United States, in China. In 2015, Xi Jinping was able to replicate the model of humanized mouse invented by Ralph Baric in North Carolina, reproduce the same model and verify that this type of coronavirus created in laboratory were pathogenic for human cells. In this period, Xi Jinping created 8 on 8 chimeric coronavirus, recombinant coronavirus in laboratory 1, all pathogenic for human cells in vitro, especially lung cells. So there is a long story on that. The last one was SARS-2 that has this new thing, fantastic in a certain way, is an admiration for this Xi Jinping because she invented, I put the insert. She studied very well the problem of immunotolerance of coronavirus in bats. She discovered the protein, the sting mechanism that can maintain download the mechanism of infection. So
you mean the spike protein?
Yeah, the spike protein.
Right.
And she discovered fundamentally the secret in which way, even if the spike protein is very toxic, very infectious, etc., it was well tolerated in bats. It was a mechanism, the sting mechanism is present also in the human cells, but in bats is down-regulated. So you don't have the cytokine store.
Right, which is still in human beings, yes.
Yeah,
right.
Yeah. So the idea of the Chinese, how we can transfer the immunotolerance of bats in humans. This is the first goal of their research.
Right.
You know, to create a probably super soldier, I don't know. But in any way. So we arrived in the moment that she decided to put an insert, and this insert must have a role of regulation of the two parts of spike. Spike 1 is responsible for contagiousness, for infectivity. But spike 2 is responsible for virulence.
I see.
So her mind said, I put some things that can drive a switch on one side, on the other side, in relation to the condition of the cell in which the virus must jacket the cell. That was fantastic. How they arrived to the furin insert, the furin cleavage site insert into the cell. This was an old story. The first patent on a furin site were deposited in the United States, in North Chapel, in 2007, 2008, discovered by the French genetician as Alexandra Code, etc. Yes. But after American people, Chinese people, Japanese people attempt to put some furin insert, but only with the 3 letter, not the 4 letter, PRRA. So it was the problem where we can take these sources. In 2018, a group of scientists in China discovered that bacteriophage was very toxic for algae in the ocean, and discovered this sequence that controlled the furin cleavage site. Yes. So they take it that it was published by Sang Gao, who's a professor of bioinformatics in Nanjing University. After 1 year of work in 2000, when he's starting the epidemic in Wuhan, this professor Sang Gao published only one publication that now you don't find at all in China, that published the origin of the furin cleavage site from this bacteriophage of the ocean. So fundamentally for Xi Jinping, he has the good model, take that, I put into, because it's a biocompatible, it's a bacteriophage, no? It's another virus, it's very compatible, you can enter into the space and put in. What happened with this type of mechanism? This is a story that Xi Jinping learned from Rotten. Rotten was the first who said,
in Amsterdam,
in the hybrid, we can create a mechanism of zipping mechanism. So
like a zipper, you mean? Yes.
As a 2 zip, 1 zip in one side on S1, other zip on S2. A furin cleavage site to create this type of mechanism. When you look on the wave on the clades that are arrived in all this period of pandemic, you see that from the beginning there were 2 variants of the virus who escaped from laboratory of Wuhan.
That's my question. That's my question. Yeah. How come this escape took place at this time? Was there any explanation? Because you have said that there were 2 laboratories in China, one in Wuhan and one in Harbin.
Absolutely.
And there were viruses escaping from the two labs pretty much at the same time.
Yeah, no, it's interesting. Yeah, absolutely. Because you have only 2 hot spots of the infection in Wuhan in January, starting, etc. In February, it was in Harbin. In Harbin was the Institute of Virology for Influenza, founded under the aegis of the FAO, Food and Agriculture Organization, the United Nations. And in 2013, they created the first hybrid, the chimeric recombinant hybrid for influenza virus from swine flu and the human. They create another very toxic virus. So, and
from the pigs,
chief,
you mean? Yes, from the pigs. Yeah.
And the chief in Harbin is a woman of the same age of Xi Jinping. And the people of Harbin were in the administration council of Wuhan. So all the laboratory work in a sort of a mirror system in which fundamentally all the samples stored in Wuhan, they were stored also in Harbin.
Yes.
The American people were aware about that because they have done conference in Wuhan, in Harbin, the same group, Rob Barich and the other. They go there, they do big conference on this type of things. So from the American institution, it was clear that Harbin was a second P4 laboratory on which they attempt to do different things, different studies, but always to create chimeric recombinant viruses. The hypothesis in which way he escaped from the laboratory. There are some evidence from the spy satellite of the United States that in October, at the beginning of October, 6/14, it was an hazardous event in the laboratory of Wuhan. It was an evacuation of the laboratory, no people there, no trace of infrared sign of human being there for 10 days, and no sign of mobile phone, telephone into and around the city.
So you're talking about October 2019, right?
Yeah, absolutely, absolutely. And you remember that 1 week after October 17, starting the military Olympic Games in Wuhan, in which more than 10,000 people arrived there.
Including many American athletes, yes.
Yeah, absolutely. And if you look on the story reported by many journals, many athletes that coming from Wuhan in Europe, for example, in Norway, in Spain, in Italy, in France, etc., but also some people in the United States, refer to have some pneumonia that were treated by the authority. Yeah, by the authority, there's a flu, okay, you go in Asia, you take a flu, it's normal to take a flu, etc., etc. And it's strange that no one has done control on antibodies on this type of people. That was very easy to do that if. So it's possible that one part of the circulation of this virus in a very discreet way circulating in Europe in November, but only some spots that are not well classified. In reality, what happened is possible that the virus escaped from the laboratory for an accident. But when you look on verified by different commission in 19, at the beginning of 19, it's very difficult that. It's very difficult.
You mean in the beginning of 2020,
yeah.
You mean?
Yeah. And you consider that only this type of virus escape is strange. If you have a biobank where there are concentrated all the different pathogenic virus, remember that Canada give all the pathogenic virus as Ebola, as Marburg, etc., to the Wuhan laboratory. So there were many pathogenic viruses there. Strangely, they escape only this type of coronavirus with the two variants, with the two clones, with the small difference in the amino acid. So L and S, that Chinese described very well in a different publication. In Harbin was the same thing. Or the same thing.
Yeah. So also, any virus escaped. The same type of virus escaped.
Yeah. The same. It's strange. This coincidence is strange. Yeah. But you must consider that at the epoch, China was very aggressive on 3 targets. One target was Taiwan. Because, and the military, they developed the theory. We want to arrive in Taiwan with the war non war using biological weapons. Is written in many documents of the military people officially in China, starting from 2015. The strategy on Taiwan.
Right.
It was the second strategy to stop the rebellion of students in Hong Kong, protesting. And the third, to control the population of Uyghur in the northeast of China, that are the only Muslim enclave in China. They have a tend to repopulate this region and to rebalance, the quantity of population, etc. But fundamentally, they want to eliminate that. So there were internal motivation of China to develop a biological weapon that can give targeting specific population. And it was written also in their program to have ethnic biological weapon. Ethnic biological weapon is written in their document. So we don't discover nothing that people don't know.
Well, I remember you saying that in a way, China had borrowed that concept from Israel, among others. The idea of developing biological weapons that can be ethnically targeted.
Absolutely. It was the great program of Xi Jinping on the fusion, so civil military fusion. So each citizen in China can be a military soldier, a soldier. It's the same model in Israel that we had it. And on the other side, on the research, they decided that the military environment must, with the Academy of Science, must pilot all the research, biotechnological research, but not only that, in China. So with a vertical system, as in a certain way is done also in the United States, because DARPA, that is the Agency for Research and Development for the Pentagon, work financing more than 60% of the research in university academic environment. So China attempted to use this type of model to adapt naturally to its system and to create its bioweapon arsenal as the other country. Remember that China is the only country who never signed, ratified the Convention Against the Biological Arm. She adopted, no, she said, okay, I agree, but never ratified. It was the only great country in the world who never ratified this. And this is the reason that American people go there to work on some biological at-risk research. And second, when they create in Harbin, why in Harbin? That is in Northeast,
Harbin, you mean, yes.
Harbin, yeah. Because it was the center of the lager of the Japanese, of Japan in the Second War, China, Japan. And there were put the lager as the Nazis lager with the famous doctor, Japanese doctor, who do experimentation on humans.
Yes, on Chinese.
So China remember, yes, China remember his history and put this laboratory in front of Japan, in Manchuria. So you are a great expert on that. So there are many political and historical motivation on that.
Let me tell you, let me ask you this. According to you then, this virus is not really meant to kill in a very large scale. It's more of a virus to weaken a population, right?
Yes, yes. And it was described in the political document of the PCC and the military academies. They said, what we want to disrupt the social economical balance of a country with the type of bioweapons. So we arrive, we can dominate very easily because the people don't have time, and we target only population that fundamentally are not useful to rebuild the country, as old people.
Right.
And this is the reason that one of the activity of the virus go on the second part, in which there is the other receptor that furin activate, that is the TMRSS2 receptor, that is linked to the androgenic receptors.
In other words, to fertility.
Yeah, absolutely.
Now,
absolutely.
Let's go to the second stage of this discussion, which is the vaccine. Because as you know, in the document that has been released by Veritas from the Pentagon, it seems that the virus and the vaccine are two things that are together, basically.
Yes.
The recombinant virus carries its own vaccine, and we know that at least at the military level, the vaccine was instantly available because it had been developed together with the virus. Now that we are talking about mRNA vaccines, RNA messenger vaccines.
Yes.
What do you have to say about these vaccines? Because a number of experts have warned about the great danger to human health from mass vaccination.
Yeah.
What do you think about that?
Okay. The research in 2007, Professor Ralph Dennison Vanderbilt, in Vanderbilt. Okay. Working with Ralph Baric, he created the first antiviral drug, remdesivir, for Gilead against, in 2007, you imagine, against coronaviruses. And he developed the idea to create a vaccine against the coronavirus using mRNA. So the first laboratory, great laboratory. Matt Dennison is a great expert on that. But the idea to create this vaccine, it was to create a vaccine for soldiers, for military environment. If you have a bio warfare, an attack on coronavirus, we have a vaccine that can be given to our soldier only for a short term, 3 months to cover it and to win the war.
Correct.
So we were in a stupid things at the end with the panic in the United States and in Israel that someone as starting a biological war, they decided in a short time to use potentially anti-bio weapon as antivirus, virus antivirus. And we are in this stupid situation. Many eminent people alerted also Israel that was not a good solution because this was experimental, not made sure to control very well. And mRNA drugs are very complex to control into the cytoplasm of the cells, especially human cells. So, but people take this option in Western countries, they take this option to do this mass experimentation on humans with this fear that the charge on the population. And now we see the consequences of that because we have a lot of side effects, not efficiency of this type of vaccine because they decayed during the time. In 3 months, at the 4th month, for example, Pfizer vaccine has a 40% of activity, and at the end of 5th month, 17% of activity. So the other 3 months, people are not covered at all. This was known from the research people from the beginning. And this doesn't not explain to the population, also on the mass media. But the other aspect that now we have discovered that this type of vaccine who create fundamentally a small COVID with this small spike protein,
Yeah, that's what we see in Europe now. It's spreading like fire. You know, people are catching COVID by the millions.
Yeah, absolutely. And they do a depression of the natural immune system that we see now between people who are in front of people who are not vaccinated or people who can take the COVID and then they have a strong response on the antibody response. We see that in the population clinically is evident, this type of things. So this solution was bad, costs a lot. Only the great company take a lot of money, and we have the need to change this pathway because it's not the good pathway to respond to this type of recombinant chimeric viruses.
But do you agree with people like Montagnier and Van den Bosch and McCullough and Malone, who are all eminent experts in their fields? Do you agree with them? I could mention 20 more, that this could cause mass death among the vaccinated populations. I'm talking about the mRNA vaccinated populations in the coming years.
If you look at what happened in the study of Jansek in MIT in Boston, who showed that in the copy choice of the SARS-CoV-2, you can copy fundamentally part of the segment of RNA not in a random way. There is a sequence for that, and this part can be inserted into the human DNA. He repeated 2 times the research in laboratory, in the experimental laboratory. There are other groups who are confirming that. So SARS-CoV-2, a difference of the other coronavirus, has this selective mechanism of copy choice in which you can take a part of the segment that can be copied in DNA and transferred into the human DNA. Okay, first. Second, with this type of vaccine, that are not vaccine, that are transfecting agents. This is the true name.
Which, by the way, violate both. They violate the UN protocol.
Absolutely. And so the risk was they can attempt to escape with the nanoshell. They attempt to escape to the immunosurveillance of the cell. When they enter into the cytoplasm, but you have a sensor, RNA, a DNA sensor. So the message that was into attempt to escape to that and to arrive on the DNA and to link.
Yeah.
And because you have a support, a nano support that is very stable, except the liposome can be degraded. But in the other variant in Moderna is not a nano liposome. It's a thing that is stable. So remain attached on the DNA. So the risk that we can have to activate some oncogenes near the area in which is attacked the information, or activate some autoimmune reaction in the future, or when this protein degrades, the spike protein produced by the vaccine, this protein cannot be completely degraded, as in SARS-CoV-2. You have the spike protein you find in the interstitial of the different organs, can be transformed in a prion protein.
That's a prion here.
Yeah. So
when you say prion, people think of mad cow disease, you know?
Yes, absolutely. But the problem is that fundamentally on the biochemical, biophysical basis, all these things can happen. Can happen. And there are some studies also from Sloan Kettering in New York, done a study on this type of things for the oncogenes. There are all the group that work with the Prusiner in California, but other group in New York also work on prion disease that show that there is a part of the spike protein that has a domain that is linked to the expression of a prion disease. So there are many elements. And after we see what happened now that you have the long COVID patient, people all recovery, but you have now the post-vaccine patient.
Yes.
Who have a similar syndrome that long COVID or post-COVID. And in some countries, as the UK, for example, they created more than 30 centers for long COVID. But now they begin to take a post-vaccine syndrome. So it's a reality. In other countries, in Italy, for example, people say, no, it's only rehabilitation, nothing to do with that, etc. They deny a clinical evidence that these people have fog into the brain, they have a neuromuscular asthenia, and we begin to show that these people beginning to develop some sort of immunodeficiency syndrome that was similar as HIV. And this is this is a good problem for the healthcare system in Western countries. We have the need to discuss seriously about that personally.
To sum up, I would like to say that clearly, not only have the mRNA vaccines violated UN protocols,
absolutely.
And therefore governments which have been trying to enforce vaccination, make it compulsory, as in Italy, or almost compulsory, as in France, they have obviously been violating their own laws, and they have been endangering people without giving them any guarantee or protection, because the governments are not taking any responsibility.
Absolutely. When the protocol were activated for this type of vaccine, it was a phase 3 of a protocol, a clinical trial. So they asked to all people to sign the consent, informed consent. But an informed consent was incomplete because the people don't describe the side effects that were possible to do. In this informed consent, you find only the signature of the patient, but never the signature of the doctor, or there is no signature of the institution, nothing, etc. They are totally illegal from this point of view, and they are incomplete. And in any way, because these drugs works directly on the human DNA, we are in the complete infraction of the protection of the integrity of human DNA that was established by the United Nations. So, and because the side effect, the people who die also for this vaccine, we are really into the system of Nuremberg Code and the treaties of Rome. We are in this, crime against humanity and the health of people. And it's clear that personally, as a medical doctor, I see that people go up the level that was necessary and maintain the precautional level principle to say, okay, it's experimental, we have that, but the patient must be aware on that. Not tell that, no, this type of vaccine don't have any effect on DNA, are not genic, not transgenic, etc. And after all the people of WHO told after, no, they have an effect on DNA. That is crazy because...
not only that, telling people to have the vaccine again and again, every 3 months, without which you are not supposed to have any social life and you become excluded, you are excluded from society. In fact, this is what Dr. Rainer Fulmich in Germany and his international team of lawyers are doing. They are essentially suing governments for crimes against humanity.
Absolutely. And they stopped also the capability of the doctor to do early treatment for people, to stop the enter of the virus into or the replication of the virus, to stop this type of mechanism. They put many drugs.
How do you explain that they didn't want, Western governments basically denied people access to drugs, to medicines that could help in the cure and in the prevention? Why did they do that? I mean, this was very obvious in Europe particularly, and even in America, they banned ivermectin,
In Europe
hydroxychloroquine.
And it's interesting that the Chinese people from the beginning, they test hydroxychloroquine and remdesivir in vitro in laboratory. They work together very well against the infected cells, but on the humans, hydroxychloroquine work, but remdesivir doesn't work. They publish so from the beginning, so no way to discuss a lot on that, etc. It was not a fight between among companies. What is sure that the Western countries were implicated into the research in laboratory in Wuhan.
Of course.
They are co-responsible in a certain way what happened in Wuhan. In Wuhan, the laboratory was specialized only to create chimeric recombinant virus from coronavirus pathogenic for humans. It's the only job that was done there. And there were American people, English people, German people, Canadian people, French people, etc. All these type of people. Because it was a parallel research there in Wuhan to create a universal coronavirus vaccine against to protect animals for the intensive livelihood of animals as chicken, as pigs, etc. It was the Fulbright Institute supported by Bill Gates to promote this type of research on universal vaccine to protect chicken and pigs from coronavirus flu. That is a huge market. If you imagine only China, how many said. So this was this parallel research. In a certain point, there was a convergence between the business in agriculture in a certain way, with the business that, oh, why we cannot create also eventually after vaccine for humans? And today, the cooperation between the laboratory of Ralbaris, laboratory of Wuhan, continue to create a universal vaccine against coronavirus as on spike protein. They continue the money from the United States go to China. They publish. There are recent publications on the universal coronavirus vaccine, etc. But what is interesting now?
Cooperation between China and the US in many fields, and the cooperation is only increasing despite the war rumors we hear about. Now,
absolutely. Discontinue.
This is a very sobering, fascinating, but sobering and even frightening conversation. But as a sort of final question, I would like to ask you with regard to India, in particular, because we don't know whether this infection, whether this pandemic will continue in one form or another, or whether it is coming to an end. And given that the mRNA vaccine path is obviously the wrong path, as you have said, what do you suggest is the avenue, especially for India, to develop a proper treatment and in order to deal with the continuing epidemic or whatever epidemic comes in the wake of this one?
In India, from the beginning, many Indian research people, as also Professor Gua, they attempted to find some solution to stop the entry of the virus into the nose, into the mouth. So they create some spray, some product that can be used in a saline solution that can stop the entry of the virus in. This was one thing. So Professor Gua wrote to the representative of India in WHO to show that they were ready to do a protocol on that. This was one first idea in India to stop the entry of the virus. This is one thing that can work very well, because now in Israel, they created some sort of spray with the nanoparticle. They can link to a stop and neutralize the virus when you enter into the nose, etc. Very, very simple things. And because the virus is sensitive to the pH, if you increase the basic pH, is neutralized. Very simple way. Okay. The second was to attempt to stop the replication of the virus when it's entering into the cell. This is more dangerous because you have the need to stop that. And there is one way you can do that, because the virus utilizes the furin site and the furin also intracellular. The furin is linked to sort out from its niche of the zinc. The zinc finger is a sort of gate that maintains the furin into, and when reduced level of zinc is open, and the furin enter to retailor the virus into. And was one of the action of the hydroxychloroquine to increase the level of zinc into the cytoplasm of cells. Unfortunately, there are many ethnic groups in the world who have a low level of zinc, and they have a low level also of vitamin D3. So their immune system is not well regulated, is down-regulated, so the virus can enter. There is a mechanism that we have studied with the people in India, also Gua, but other people in pharmacology, that we can change the zinc with other cationic, metallic cationic, using peptide inhibitors. I will write only some small words in my book, etc., that reinforce the system of zinc finger. So the real weapon of this virus is a furin that is also a toxic protein. When it's detached on the virus, work as a toxic protein, as a poison for a snake. So the Indian people start immediately. Also, there are brilliant bioinformatician people who have detected all this type of mechanism. Now, I think there was one decision of the Indian government, because the Western country don't want to give the intellectual properties to create facilities in India to produce mRNA vaccine. They said, okay, now we begin to work on a new idea to create a DNA vaccine.
Correct.
This was announced by the government.
It is, yes,
they can be more brilliant in the sense that the target, but naturally has also ethical implication on that, because each time that you go on human DNA, you have the need to consider all this type of aspects.
Plus, I think you have to take into account the fact that if you affect the DNA, you can technically claim property rights over the DNA, which means that human beings will no longer be essentially the owners of their DNA. They will become corporate products.
Yeah, but this is the other big problem. If you use also an mRNA vaccine, you are marked with the trace of this vaccine. Fundamentally, the company can be co-owner of your DNA, manipulated with this Trojan horse who enter in your DNA. This is a big issue that has the need to be debated at the United Nations level. Otherwise, the risk is not for the country. You imagine Russia was a great master of bioweapons in the world. Russian people, long tradition on that. But first, the number of leakage in the laboratory in Russia was very poor in front of the United States. And second, they never use it as a bioweapon, even if there is a deterrent, but they never use. So some countries are more equilibrated in what they do. But the risk today is the biotech.
They take a more healthier attitude to this, yeah. Exactly.
Yeah, absolutely.
Well,
it's a question of control.
I know Russia is very tightly controlled on this score. They are very concerned about the fact that the Americans have set up a number of biolabs around Russia, in countries like Georgia and others. And the Russians are very concerned. They have called attention on that, saying, are they trying to threaten us with a biological infection?
Absolutely. And so the real problem is if the big company said, okay, because I have manipulated your DNA belongs to me, and you belong to me, because if you have a pathology, whatsoever pathology, you must take our drugs, because we know what you have done on your DNA. This is a totally unbelievable scenario, the way in which people can be controlled by big pharma.
Yes.
A big pharma now attempt to say to the country, okay, you don't have money to take in charge of the healthcare system. We offer to take in charge as a provider of services for the healthcare system. So we give the money to you, but you are the owner on that. That was happening, especially in Europe now, because Europe, you know, that is not a political unit, it's a commercial. So imagine, if you don't give money, how the European bank can work, etc. So this is a great risk on that, honestly. It's a great risk.
In France now, people are openly saying, some prominent people are openly saying that, are we going to have to pay a yearly tax to Pfizer? You know, because basically yes. Pfizer is in charge of health control.
Fundamentally, is that many, as what's happened also in Israel, Pfizer take the exclusivity of the domain and the data of the patient who were submitted to the vaccine, including the genetic data. In Italy, I remember when it was the government Renzi gave to the IBM the possibility to enter in all the database of Regione Lombardia to take all the biological data of patients who are taken charge by the healthcare system in Regione Lombardia. It was paid for that, and they continued this type of cooperation. So you create a big database, then you analyze the cluster of population. You said, okay, 20% of this population is a cardiovascular. So we can target this type of population with these drugs. We put this marker and we monitor population this way. But this is totally dangerous. It's dangerous because no people can, who watch the watcher, who control the controllers. This is the basic question for democracy, for freedom, and for the protection of the integrity of human people. This is not a question of an epidemic. There will be other epidemics, other pandemics, etc. But the way in which we decided to manipulate the human genome, because they used the Emergency Act to do that.
Yes.
Also,
they have broken all the laws. I'm talking about Western governments, you know, in particular.
Yes, absolutely, absolutely.
Well, this was I wouldn't say it was a pleasure, because it would be to put that forward, but it was extremely informative and enlightening. So I would like to thank Professor Giuseppe Tritto, President of the World Academy, President of the World Academy of Biomedical Sciences and Technologies.
Yes.
And thank the India Foundation for making this podcast possible, hoping that many people will derive benefit from it and we'll get a clearer picture of what is happening in our very tormented and troubled times.
Yes, surely, surely. Thank you very much for your support. Thank you very much,
Professor.
I enjoy a lot. Grazie. Arrivederci.
Arrivederci.